Very good morning to all of you. Myself, Dr. Surbhi, Assistant Professor in the Department of Zoology, School of Basic and Applied Sciences, Maharaja Agastyan University, Baddi. Today I am going to discuss about the mutations and some genetic disorders. So first of all, what is mutations? Any change in the DNA sequence or any mistake in the DNA sequence, it is known as mutation. It could be point mutation or multiple mutation. Point mutation means if there is a change in a single nitrogen base in the DNA. Like as you see in this very diagram, this is a sequence of normal DNA sequence, and here if this thymidine is replaced by adenine, so the DNA become mutant and it is a single change. But if the one or two of the nitrogen bases change, then it is known as multiple mutation. So these are the mutations when there is a single base pair, or you can say nitrogen base is changed in the DNA. Now, these mutations are of two types, chromosomal mutations and genetic mutations. Now, coming first to the chromosomal mutations, it occurs when there is a change in the chromosomal structure. Its shape and its structure get changed. So, it is on the basis of this, it is of four types: inversion, translocation, duplication, and deletion. Now, coming first to the inversion, when the order of a gene or in a chromosome is inverted. As you see in this very diagram, this upper one is the normal chromosome, and here the sequence of gene is A, B, C, D, and E, suppose, for example, right? And when if there is a break in the chromosome and this sequence, this very BCD, it get inverted and it become DCB. So this type of mutation is known as chromosomal inversion when the gene sequence is inverted. Next is the translocation. Here, the movement of a chromosome fragment to a non-homologous chromosome. As you see in this very diagram, here there are two types of chromosome, one in red color and another in the blue color. As you see, there is a break in the red chromosome and it is going to replace by the blue chromosome, and also a break in the blue chromosome and it is going to replace by the red chromosome, mean they intermittently cross their regions. So it is known as translocation type of chromosomal mutation. Now coming to next, deletion. If there is a loss of few bases or loss of large region of chromosome. As you see in this very diagram, if this sequence, this TAC, it got deleted, then the mutant DNA will become shorter, and it could be three triplet or it could be multiple triplets, means maybe loss of few bases or large number of bases. Now, next is the fourth type and the last one, duplication. Duplication of a few bases or duplication of a large region of chromosome. As you see in this very diagram, here TA, that is thymine and adenine, it is getting duplicated. If there is more than nitrogen bases going to duplicate, it will be known as the duplication of the large region of chromosome, and it ultimately led to the increase in the number of increase in the number of nitrogen bases, and the length of mutant DNA also increased. Now, how this happened? So first phenomena is crossing over. What is crossing over? It occurs when the chromosome they exchange their genes, when they overlap. Some genes cross over and switch their places. As you see in this very diagram, the below one, it is easily to understand. One chromosome, it is in purple color, and other one, it is in reddish or pinkish color. And see, their lower sister chromatid, they are going to exchange. As this occurs, crossing over in the purple chromosome, this pinkish region appear, and in the pinkish chromosome, there is a purple region appear. It means there is a crossing over of homologous chromosome. Now, what is non-disjunction? If the chromosome pair fails to separate properly during the process of meiosis. As you see, during the metaphase, the chromosome will become middle in the middle of the cell, and at the anaphase, they start moving towards the poles. And when it occurs, there is a less chromosome in one of the gamete. So it led to multiple disorders, out of which one example is monosomy. In this, a gamete has one less chromosome than it should be. So the result would be 45 chromosome. As you see in this very diagram, there is only one sex chromosome. And the syndrome that occurs is known as Turner's syndrome. It means missing a sex chromosome. Next is the trisomy. It means a gamete has one more chromosome than the normal chromosome, and the result is total of 47 chromosomes. As you all of you know that there are 46 chromosomes in the human being, but due to trisomy, it becomes 47. And the syndrome is known as Down syndrome. Here there is an extra 21st chromosome in this very human being. Now, what are the methods to detect these chromosomal abnormalities, or you can say these chromosomal mutations? So, first is the chorion villi sampling. So, as you all of you know, chorion, it is the embryonic membrane that surrounds the fetus. As you see in this very diagram, these are the finger-like structure originating from a membrane. This membrane is chorion, and these are the chorionic villi. And below this is the placenta. So, what we do? We take a sample of chorion, the membrane which surround the fetus. We do the chemical test and karyotyping is performed. Second is the ultrasound. It is just the sound waves that are used to generate an image of the unborn child. It is used to detect if there is an abnormality in the limbs or organs, etc. Now, third, it is amniocentesis. Here, there is a fluid which surrounds the fetus. It is taken up by a needle, as you see in this very diagram. This needle is inserted in the stomach, and this amniotic fluid is taken up. Then these fetal cells are grown in the cell in the lab, and the chromosomes are then karyotyped. But karyotyping is the arranging the chromosome due to their shape and due to their number. So in this way, the chromosomal abnormalities can be detected. Next are the autosomal disorders. It is of three types. Down syndromes, here it is a trisomy of 21st chromosome. Patau syndromes, trisomy of 13th chromosome. Edward syndrome, trisomy of 18th chromosome. Now taken up first the Down syndrome. It is the excess of 21st chromosome. And here the prenatal testing can be done for its detection, and it is a result of chromosomal mutation. One in the 900 people born with this very disease. Likelihood of having a child with the Down syndrome increases with advancing their maternal age. And its symptoms are mental retardation, upward slant to eyes, small mouth, abnormal ear shape, decreased muscle tone, and there is no cure for this very syndrome. Next is the Patau syndromes and Edward syndromes. Here there are cardiac abnormalities, they're very severe conditions, and mostly the infected infants die during the first few weeks of their life because of these very two syndromes. Next are the deletion disorders. It is also of two types, Engelmann syndrome and Prader-Willi syndrome. Coming first towards the Engelmann syndrome. Engelmann syndrome, here the symptoms are inappropriate laughter with the convulsions, poor coordination, and there is mental retardation. And the human being appear like as you see in this very picture. Next is the Prader-Willi syndrome. Here the symptoms are extremely floppy, obesity, and mild mental retardation. And the human being appears as you see in this very picture or image. Next are the sex chromosome disorders. These are also of three types, Klinefelter syndromes, Turner syndromes, and Fragile X syndrome. Talking first about the Klinefelter syndromes, here there is one X extra chromosome. The X is one extra. It occurs 1 in 1000 live births. Here the symptoms are mild learning difficulties, taller than the average with long lower limbs, Show mild enlargement of breast. Infertile, it means there is absence of sperms and it could be treated with the testosterone therapy. So as you see in this very diagram, it is a male, but it's showing the female-like physical characters. Next is the Turner syndrome. Here, there is one less chromosome, so the result is 45 number of chromosome. Here, it is very low in incidence. If the human being appears normal, there is ovarian failure, normal intelligence, short stature, and it could be treated with the estrogen therapy. Next is the Fragile X syndromes. It is most common inherited because of the mental retardation. It occurs 1 in the 2000 males, and the symptoms are high forehead, prominent jaw, and autism. And in this picture showing the Fragile X syndrome human. Next is the single gene disorders. These are of four types: cystic fibrosis, hemophilia, sickle cell anemia, and phenylketonuria. So, talking first about the cystic fibrosis. So, it is a recessive disorder. Mutation stops the production of protein in lung cells as well as in the pancreas. There is a thick mucus, bacterial infection in the lungs. A test is performed for this diagnosis that is known as sweat test. It is most common in the Caucasians, that is 1 in 3,300 people. And the symptoms are chest precautions, diet supplements, and it's shortened life at having the human having this very disorder have the shortened life expectancy. Next is the hemophilia. It is a sink-slick disorder. Here it is a failure of blood to clot, as you see in this very diagram. It is very rare in females, and the injection with the clotting factor can be used to stop the bleeding in these very patients. Next is the sickle cell anemia. Here is a mutation in the blood protein. The RBC, which are normally oval in shape, they become sickle in shape. And there are screening tests for this. It is very common in Africans and Americans. That is one case in 375 people. And the symptoms are pain associated with blocked vessel causes anemia and fatigue, and it is very common where the mosquito-borne malaria is present. And last is the phenylketonuria, mutation that disturbs the function of enzyme. It leads to the high phenylalanine levels in brain, mental retardation, epilepsy, and screening newborns. It occurs 1 in 10,000 caution births. Extremely rare in Americans as well as in Africans. The patient suffering with this very disorder looks normal and it lead low protein diet. So these are the chromosomal as well as genetic mutations we are discussed. Thank you so much. Hope you will like the video. Thank you.